The spread of the coronavirus.
We must test for the virus.
It has spread across the world.
We don't have immunity to this virus.
The number of COVID-19 related deaths in the country surpassed 500,000.
This surge is the worst they've seen since the pandemic began.
Follows a big announcement this morning. Pharmaceutical company Pfizer says its vaccine is 90% effective.
We've all lived through it and are now approaching a return to our normal lives. But the thing that changed it all was the COVID-19 vaccine. How much do you really know about it? The COVID-19 vaccine took a year of hundreds of thousands working together and many different failed trials. Let's explore this path from an unknown virus to a nationwide distributed cure. To begin, most vaccines utilize one of two different forms of a virus. One type of vaccine injects a dead virus into the body, allowing an immune response to the virus without risk of actual infection. Why didn't scientists use this method for COVID-19? Dead or inactivated virus vaccines don't create a lasting immune response, according to the U.S. Department of Health and Human Services. This type of vaccine can be seen in the flu vaccine. Another type of vaccine is live attenuated, or a weakened virus. By injecting weakened viruses, our body can create a lasting immune response. If this was used for the COVID-19 vaccine, however, weakened immune systems could actually develop the exact disease that the vaccine is targeting. Two examples of this type of vaccine are the MMR, or measles, mumps, and rubella, and the chickenpox vaccine. The third type of vaccine, and the one that we'll focus on, is the mRNA vaccine. First, we need to talk about spike proteins. The spike proteins that you see all around the COVID virus molecule are actually what alert the body to trigger an immune response. Let's take a closer look at these proteins. In our bodies, proteins are made by ribosomes using mRNA as a template. mRNA stands for messenger ribonucleic acid. You can think of ribosomes kind of like a translator, translating from the nucleic acid language to the amino acid language of the proteins. Usually in our bodies, mRNA is translated by polymerases from the DNA language, which is another type of nucleic acid. There's a misconception that the mRNA vaccine changes our DNA. While some viruses have the ability to alter the DNA in cells, this vaccine doesn't actually contain a virus, so it can't change your DNA at all. For this type of vaccine, mRNA is made in a lab and packaged into a vaccine. When this mRNA is injected into your body, your cells actually take it in, using their internal machinery to produce the COVID spike protein on the surface of your cells. Once this spike protein is on the surface of a cell, immune cells in your body come to form what's called an antibody. These antibodies act like a reference manual for how to fight a certain virus. We store these reference manuals in our bodies, creating a sort of immunolibrary. So if we're ever infected with COVID, mumps, or the flu, our body knows how to recognize and fight the infection. mRNA vaccines can be produced faster than other vaccine types, but can be more difficult to scale because the mRNA is unstable, which is why it must be packaged and stored at low temperatures. Despite this difficulty, however, scientists are currently researching other applications of mRNA-based vaccines, such as applications for treating cancer with immunotherapy. According to a 2021 journal article by Miao Zhang and Huang, the first therapeutic cancer vaccine was approved by the United States FDA in 2010 for treating prostate cancer patients. This technology has been around for years, but scientists from all over the world came together to push for a solution to a problem that literally stopped modern society in its tracks. By working together and through the use of science and technology, we have gone from being isolated and alone to slowly working our way back to normal. Thank you.